Hello and welcome back to PBH435. Today we're going to be covering our lecture on Chapter 4, the nervous system. So it's important that we understand the functions of the nervous system, and the reason that we go over them here in our class on health studies of dangerous drugs is because basically this is how drugs interact with our body. They affect our nervous system. Our bodies in general like to maintain a condition called homeostasis. This may be something you've encountered in other kinds of classes, but for those of you who hadn't, basically our bodies like things to remain the same. Whether we're talking about temperature, the water content of our body, glucose concentrations, acidity, our body has a certain standard that it likes to maintain. A lot of us are familiar with the example of body temperature being 98.6. If we start to get too hot, our body starts to sweat to cool off. If we start to get too cold, our body starts to shiver to warm up. So our body likes to maintain that internal standard within certain limits. Now, when we start talking about the use of drugs, many of them are going to influence that status of homeostasis in some way. A great example of this is one that some of us may be familiar with. It involves consuming alcohol and the need to urinate. So alcohol inhibits the release of vasopressin. Vasopressin is an antidiuretic hormone. So after alcohol is consumed, more urine is produced. And we see actually some interesting effects on homeostasis depending on how accustomed a person is to drinking. To someone who doesn't drink very often, compared to someone who doesn't drink very often, a heavy drinker is going to produce less urine because of that effect on vasopressin. Now, during alcohol withdrawal, heavy drinkers will again see that difference response. They'll see increased vasopressin release. So that relationship is about homeostasis. It's about the body trying to maintain a certain normality in terms of its water level. We're going to be talking about a couple of different components of the nervous system today. So we're going to be digging into some details. Again, for those of you who've been through biology classes or classes that might be related in other topics, some of this may be review, but it's important that we all understand it because of most of the drugs that we study are going to be affecting the nervous system in one way or another. So when we look at the nervous system, we do have two major types of cells that we see. We see neurons and we see glia or glial cells. We'll talk a little bit more about both of those. So talking a little bit more about neurons. Neurons are those primary elements of the nervous system that are going to analyze and transmit information. We'll talk a lot about how that actually happens. But basically, before we begin, we're going to break those neurons down into their individual parts. And for neurons, we've got 4 defined regions that are important: the cell body, the dendrites, the axon, and the axon terminal. So the cell body, pretty self-explanatory. We'll see an image in just a moment. Dendrites, now the role of the dendrites is to receive information or to receive signals. And then the axon, which does the exporting of the signal, and then the axon terminals. So we'll look a little bit more about those in a moment, but there are actually more than 100 billion of these in the human nervous system. So for each of us, these 4 major regions occur over 100 billion times in each of our nervous systems. And again, if you're looking for a way to remember, dendrites receive and the axon exports. Let's look a little bit more into how that works. So here's the schematic of the neuron with those major parts we talked about. We've got the cell body right here. We've got the axon. It's inside that sheath right there. Axon terminals down at the end. And then the dendrites back here towards the left. So in each of these, for each of these 4 major regions, you can see how they fit together in this schematic of the neuron. Our other major type of cell are the glial cells. This is actually from the Greek word for glue. Glial cells are found around neurons and they provide them with support and protections. Actually, 90% of the cells in the human brain are glial cells. They form myelin. Myelin increases the information processing speed and efficiency of neurons. Have you ever heard about the chronic disease of multiple sclerosis? What we see in multiple sclerosis is an issue with the production and sustenance of myelin. So we see deterioration of the myelin sheath over time, and people who are experiencing advanced MS, they have difficulties with that information processing, both its speed and efficiency, and they can end up with motor control problems because of that. So until recently, we thought that the only role of glial cells was that structure and support, and we've actually recently learned that glial cells are actually able to communicate with other glia and with neurons. So this is definitely an emerging area in science. Another really important role of glial cells is to create the blood-brain barrier. We're going to talk a lot about that here in class. Basically, its function is to protect the brain from toxic chemicals. We want to make sure in many ways that our brain is protected from the things that enter our body, and that's what that blood-brain barrier will do. In order for a drug to actually have an effect on our brain, to become psychoactive, then those molecules have to be able to pass through the barrier. And we'll talk more about that and exactly how that would occur. So neurotransmission, this is that process that information gets passed from one neuron to the next. This is one aspect of that nervous system function that is really important to understand the effects of drugs on the body. So we're going to talk about an event called an action potential. And the best way to describe this is that electrical signal that gets transmitted across the axon. If you think about a neuron at rest, there are going to be more potassium and negatively charged anions inside the cell and more sodium and chloride ions outside the cell. So the ion channels are going to open and close. An action potential is generated. The neuron is depolarized, and then the message is passed along. So we've got that resting potential where there's that uneven distribution of the ions, and then the ion channels open, and we see electrically charged particles moving inside the cell, and then the neurotransmitters are released. So I've got a quick video that I want to show you that actually animates this, might be helpful in understanding that action potential event.
That is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus changes the permeability of the cell membrane to sodium and potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal, called an action potential, that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated sodium channels to open, and sodium ions rush into the cell. The cell becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close, while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The cell returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell, restoring the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting potential distribution of sodium and potassium ions. Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Okay, so if you've ever heard about an electrical signal moving through your nervous system, that's how that actually works. And remember, you have more than 100 billion of these neurons in your system, and they're constantly doing that all day long. Whether you're seeing or breathing or scratching your nose, all of those events involve millions of electrical signals moving through your system, so millions of those action potentials. We'll continue to talk about those throughout the semester because, again, this is how we see the nervous system disrupted by drugs. Okay, when we talk about the nervous system, we actually have three separate components, and they're named differently, all named nervous systems. So one overall nervous system, three categories: the somatic nervous system, the autonomic nervous system, and the central nervous system. So the somatic nervous system, this mostly covers sensory information coming in and then motor information going back out. So our voluntary actions, you move your hand away from the heat because it's hot, that sort of thing. The autonomic nervous system, this is the involuntary function of the body. Things like breathing, your heartbeat, things that are not under conscious control, things that are involuntary functions. And then finally the central nervous system, the brain and the spinal cord, the components that most of us are probably most familiar with. So a little bit more about the somatic nervous system. So the somatic nervous system is going to control those voluntary actions. If you're fidgeting in your seat while you're listening to the lecture, or maybe you're walking on your treadmill while you're listening to it, those are all voluntary actions controlled by the nervous system. It's going to be the part of the nervous system that carries sensory information, temperature, sight, smell, sound, into the central nervous system, and then also carries that movement information back out to the peripheral nerves. So when we talk about that information being carried, we're talking once again about action potentials. And in this case, the particular neurotransmitter that we're talking about when we talk about movement is acetylcholine. So another quick video to show us how that works. Here's the information.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So that's how that process would be happening if you were moving your hand or moving your legs or moving any kind of muscle fiber in your body. Here we see the end of a neuron connecting to muscle fiber, and we see that same process of transmitting information along the ends of the axon terminals. Okay, so that's our somatic nervous system. Next, we're going to talk about the autonomic nervous system. So the autonomic nervous system, this is all of our internal and involuntary functions, our heart rate, our blood pressure, all those things that we can't control. Now, we do see many important psychoactive drugs that are going to affect the autonomic nervous system. When we think about drugs that suppress respiration, breathing being a function of the autonomic nervous system, we can see how this particular system is one that we especially worry about, because if we see heart rate or respiration begin to drop, we're talking about somebody's life being in danger. So the autonomic nervous system actually has two branches, the sympathetic and the parasympathetic branch. And these sometimes act in opposition. So just because we know it's involving the autonomic nervous system doesn't necessarily mean that it's something that involves decreasing heart rate or respiration. It may actually be increased as well. All right, so finally the central nervous system, the one that most of us are probably most familiar with, the brain and the spinal cord. Of course, these components of the central nervous system have many, many functions. The core of our personality, integration of information, learning, memory, coordinating activities, our judgment, emotions, all of these things are based in the brain and the central nervous system. And we are going to be talking all semester long about drugs affecting these components of the central nervous system as well. So digging into the brain a little bit more in detail, we're going to be looking throughout the semester at specific chemical pathways in the brain, ways that drugs can either interfere or amplify certain chemical pathways. So we'll talk about a few. We'll start with dopamine. That tends to be one that people are familiar with. Dopamine actually has two separate pathways in the brain, the mesolimbic and the nigrostriatal. So when we talk about a pathway, I'll take a look at our brain diagram here. We are actually talking about a path from one part of the brain to the other. So if we look at the dopamine pathways, we're looking at the path from the ventral tegmental area to the nucleus accumbens, or from the substantia nigra to the striatum. So we can see how there are actual pathways. When we talk about this, it's not a theoretical thing. It's an actual way that action potentials move from one part of the brain to the other. All right, so back to dopamine. We hear about the dopamine reward system. We talk about things that making us feel good, wanting us to make us do them more often. And it's possible that this is part of how the reward system of drug dependence functions. We'll be talking about that throughout the semester. When we talk about that other dopamine pathway, the nigrostriatal, now we're talking about a pathway that's involved in Parkinson's disease. So one of the things that we'll see as we move through this information on chemical pathways and neurotransmitters, this is all very new and emerging science. And you'll see the word proposed and expected often. There's a lot that we don't know, and there's some very exciting things that we're learning. So we don't know everything there is to know about the brain. We've barely started to scratch the surface. But what we are starting to learn is very exciting. Okay, so moving on from dopamine, norepinephrine also has multiple pathways. Norepinephrine, here we're talking about arousal and attentiveness. It also is expected to have some relationship with food intake and appetite, and we see it related to stimulant drugs as well. Our next neurotransmitter, serotonin. This is one that we talk about being related to depression. A lack of serotonin has been associated with depression in some studies, and we think it may also have a role with impulsivity and aggression, and then also possibly with control of food and alcohol intake. We do know that hallucinogenic drugs such as LSD definitely influence serotonin pathways, and we'll be talking more about that during our section on hallucinogenic drugs. We talked a little bit about acetylcholine already, its connection with the neuromuscular connection, and we know that it's also involved with Alzheimer's disease. And if you think about some of the symptoms of that disease, it should follow kind of naturally that it's also associated with learning and memory. And then finally, endorphins. You may have heard of endorphins when we talk about the runner's high or the positive effects of exercise on the body. And endorphin is actually a naturally occurring opioid-like chemical. So when we talk about our opioid drugs, we'll see how those pathways are related. So endorphins, which occur naturally in our body, play a similar role when it comes to pain relief. GABA, this one we'll find almost everywhere in the brain, and we'll talk about it in relationship with many drugs. And then the same thing with glutamate. So those will come up with many of the different drugs that we talk about this semester. Okay, so here's a summary of the neurotransmitters that we talked about, the central nervous system changes that they can be involved with, and then the drugs of abuse that they're associated with. So for instance, our neurotransmitter is dopamine. We know that levels of dopamine are related to changes associated with euphoria, agitation, and paranoia. And then we also see the neurotransmitters in the same pathways being associated with drugs of abuse such as amphetamines and cocaine. So you can see how those dots start to come together to form a pattern, and these will be patterns that we come back to throughout the semester as we talk about the effects of drugs on the central nervous system. Okay, so when we talk about the parts of the brain, here's a quick graphic of what we'll actually be looking at. The brain itself, many components, and different drugs will have different effects on different parts of the brain. So those neurotransmitters that we just went through, that long list, those are the ways that messages are passed along through the nervous system. And the whole process happens very quickly. So in order to allow those messages to be transmitted, the precursors for those neurotransmitters are found circulating in the blood supply. And the first step in the process is actually uptake. So this is when those precursors are taken up by the cells around them in order to create the neurotransmitters that the process will need. The next step will be synthesis, where those precursors are actually made into neurotransmitters through the actions of enzymes. So here's an image of how that would look. We have our precursor molecules and fragments, and then the enzyme that helps with the synthesis. They bind to the enzyme, and then the transmitter molecule is created. And here's an example of how that would actually look with dopamine. So our fourth step will be storage, where the neurotransmitters are stored in the vesicles. We saw that in the video on the neuromuscular junction. And then release. When that action potential arrives, that electrical signal traveling along the cells, then the neurotransmitters are released into the synapse, or that space between the nerve cells. And then binding, so the neurotransmitters are now on to the next neuron, attaching to the dendrites, and they can continue to have their effects after that. So we've got storage, release, and binding, where it crosses the synapse between the neurons, or between the neuron and the neuromuscular junction. And then metabolism. Once the signal's been sent, the neurotransmitters are no longer needed, so they may return to the neuron, or they may be metabolized, and there may be metabolites that are left in the bloodstream, and that's something that we'll talk about more later as well. Okay, let's look at a couple of examples of how drugs can become involved. We've gone through a lot of information on the nervous system, its components, and its functions. So let's talk about how drugs are actually related to this. So we went through our list of neurotransmitters, those important ones for cell function, everything from cell function to emotion. Now, drugs can actually alter whether those neurotransmitters are available in the synapse or not. We went through all those steps in the process of neurotransmission, and drugs can impact and affect all of those. So it might affect synthesis, where the neurotransmitter is being created. It might affect storage, release, and uptake, where it's crossing the synapse, or it might affect metabolism. An example of this, antidepressants. Many of these block the reuptake of neurotransmitters like dopamine or serotonin, and because it's blocking the reuptake, then it's actually increasing baseline dopamine and serotonin levels. And we suspect this is why those antidepressants are actually affecting mood in the people who are taking them. Okay, so there's one example, altering the availability of neurotransmitters. Another way that drugs can affect the nervous system is by direct action on the receptor. So the receptors, the dendrites, Rather than receiving messages from the neurotransmitters, it may be receiving messages from the drug. So the drug is acting as if it was a neurotransmitter, and it's activating the receptors as that neurotransmitter would. Or the drug may actually be an antagonist, where it's occupying that neurotransmitter space and preventing it from activating the receptors that are key to it. So it could be taking the place of the drug, or it could be blocking the place of the drug. And these are just a couple of the ways that drugs can act on the nervous system. So because of all the interactions between drugs and its effects on moods, we've begun to wonder whether the neurotransmitters themselves are what's really responsible for behavior. And this isn't a question that's new to the 21st century. It's not something that we're just trying to answer now that we have ways to scan the brain. We've been talking about these kinds of chemical theories of behavior for millennia, really. From ancient Chinese philosophy to ancient Greek medicine, we've attempted to explain people's behavior based on levels of chemicals in their body. A current one that's actually gaining some traction has to do with theories of mood. It's called the monoamine theory of mood. And we're talking about those same neurotransmitters again, dopamine, serotonin, norepinephrine, and we suspect that too little activity in these systems can cause depression, and too much can cause mania. But we're not able to explain all of behavior, even based on this particular theory, which again is it's been fairly well supported at this time. We haven't been able to come up with a single theory or a single kind of biochemical test we can run that really explains mood. And also we're not able to find a similar conclusive theory or test for drug dependence. And even when we find things that seem like they might be important biological markers, we don't really see those holding up in an experimental setting. So we still definitely have a lot to learn in these particular areas. So just two more things that I want to touch on before we wrap up our lecture for today. We talked about brain imaging. As we continue to learn about the brain, we're learning more and more ways to basically get images of how the brain is working and what it's doing. Two important ones that we'll talk about this semester are PET, or positron emission tomography, and then the functional MRI. So a PET scan actually gives us a direct measure of brain activity. Now, to make it work, though, we actually have to inject radioactive materials into the brain. And we can see how the brain is acting at the time that the action is occurring. So some really interesting ways that we can start to learn about brain function and activity, but it doesn't tell us anything about brain structure while it's occurring. And then another tool that we're using is the functional MRI, functional magnetic resonance imaging. Now this one, we actually can see real-time changes in the blood flow in the brain, and it's not invasive, so we don't have that problem where we need to inject radioactive material. But this one, does not give us any information about brain anatomy while we're looking at it. So we're beginning to piece together the puzzle. We're learning more and more about the human brain here in the modern scientific era. We still don't have a single perfect tool, though, that gives us all the information that we'd like to see about the brain. So that does bring us to the end of our lecture for today. Please continue to keep up with the study guides and the Learn Smart modules, and I will see you next time for our lecture on Chapter 5.